All right. Well, I'll tell you what, the two presentations we heard this morning, I thought were outstanding. And both of them had me thinking, oh no, how am I going to follow that? Specifically, one of the points that Greg made, if I remember correctly, is that there are a lot of people who believe what they believe because of emotion and not because of science or rational thinking. And I thought, well, I'm not sure that my specific presentation has much to say to that audience because I'm really going to take a didactic approach and explain to you in the best way that I can what a randomized controlled trial is, as you can see on the title slide there, why we need them and can we trust them. And then Rick, you had me thinking about my patients, many of whom have died in the last 2 years. And that was not easy. And it's not easy. This approach to thinking about trials and answering questions, in this approach, people are numbers, right? People are data points. But please don't let anything I say today lead you to think that that's what I view my patients as, because they're not. They're people. And in fact, I don't go into it in this presentation, but there are very important safeguards in place to ensure that when we conduct clinical trials, we do respect those people who are participating. We don't view them as just numbers, as just data points, but in fact as individuals who are contributing to this mission to gain knowledge, which will then in turn help other patients in the future. All right, I have disclosures as well. I tried to be exhaustive here, although I didn't provide as much explanation, I think, as you heard from Greg. I received funding from the National Institutes of Health, so my research is federally funded. I do have a number of industry relationships as well, so if that's important to you, I want to be very transparent about that. I've listed all three of my current relationships. The first one is giving me funding to do a research study that is not interventional, it's not a randomized trial, and it's not related to COVID, but I want to disclose that. The second is a drug company that makes sedative agents, and I've consulted for them in the past. It's no longer a current relationship. And then the third is a company that's making a novel device that's intended to get patients off the ventilator faster. And so that could eventually relate to COVID. Right now, it doesn't, but it might in the future. And for them, I serve on an advisory panel. So I have no actually financial investment in any of these companies, but I do have an academic relationship. And then actually, on that point of academics, I think it's really important. In medicine and in academics in general, there's been a tremendous shift over the last 10 to 20 years at being more careful and transparent about disclosures. But one thing that disappoints me is that we're not actually very thoughtful about probably the most important investment that we have, which is our academic reputation. So all of us who do research, and I think Greg and Rick would agree, this is our whole life. I mean, our whole career is determined, and the success of that career is judged based on whether or not we fail or succeed in our research. Now, and failing and succeeding in research doesn't actually look anything like you might think. It doesn't matter if the trial was positive or negative, if the treatment that's being studied did what you think it would do or not. That's not relevant. What's relevant in terms of your academic success is whether or not you design a carefully thought out trial and you successfully conduct it, and then you publish it. And then based on that, you're judged. And then you're judged by your peers who determine whether or not your work was valuable and is worthy of publication. And then they, in turn, decide whether to give you more money to do more research. And so, we spend a lot of time talking about our financial relationships, but in my mind, what you need to understand most is that my academic reputation hinges on whether or not I do work that's considered rigorous and successful. So does that lead to bias? Absolutely, it does. And I want to describe to you the steps that we take to try to safeguard in light of that bias. So what are we going to talk about? 3 questions. And raise your hand if you're in a medical field. Okay. So I'd say maybe 20% of this room. I don't know how many online are. I'm relieved that it's not a higher percentage because I really designed this presentation with those who are not in medicine in mind. Hopefully, I actually really like to hear your feedback afterwards. Let me know if I made this too simplified. My goal was to make it accessible and understandable to people who aren't in the field of medicine. Let me know later, not when we're recording, if I succeeded. So I want to answer 3 questions. 1, why do we need randomized controlled trials? And I'm going to use this abbreviation, RCTs. What I mean is a randomized controlled trial. 2nd, how do they work? And then 3rd, are they trustworthy? Okay, and I actually gave this presentation to my kids last night, and one of the pieces of feedback they gave me is, well, Dad, you didn't explain what a randomized controlled trial was early enough. I ignored that feedback, so I actually answer that in the second part of the presentation. Again, let me know later if that was a mistake. So why do we need randomized controlled trials? So, moving a little bit faster. We've already heard this discussed. There are different levels of evidence. I think, Rick, you talked about the grade approach. That's actually up here in the sort of synthesis stage. You take multiple studies, and the idea is that looking at multiple studies is better than looking at a single study in terms of trying to discern truth about, in this case, something medical. This methodology could be applied to non-medical fields as well. But in this context, we're trying to learn, is a vaccine effective? Is a treatment effective? Is some device effective and safe? These are the things we learn from individual studies, but then a higher level of evidence is to take that data from multiple studies and combine it together. And I'm not going to go into the details here, but there are different ways to do that, and they're considered sort of exceedingly more rigorous. But I want to focus on this bottom part of the pyramid, what I would call the foundation. All other knowledge in medicine in this context is based on a foundation of individual studies. And when you dive into those individual studies, you see there are lots of different types. And each of those types of studies or those study designs has a different level of quality and of rigor, and therefore is viewed as more or less reliable than the others. So in randomized controlled trials, which is the focus of my presentation, is at the top. So when it comes to individual research studies, a randomized controlled trial is considered the highest quality possible individual study that you can do. Well, why is that? I'm not going to explain to you the details of what each of these other types of study designs look like, but I want to point out some limitations that are common to all of them. And these are limitations that randomized controlled trials are designed to address. Before I move on, though, I do want to point out that at the very bottom, the lowest quality of evidence, what's listed here in this figure, is case reports and case studies. A case report is just what it sounds like. Somebody sees a case, a single case, and then they report it. And that has a valuable place in the literature. There are certain things that we can learn from those cases. A case series is just a series of individual cases put together and reported. There's another form of evidence that is at this level or even lower, and that is just anecdotal observation. I don't want to pretend that that's not evidence at all, because it is. When a clinician is taking care of a patient and they say, I'm going to do A, and then I'm going to observe whether B happens or not, that is a form of evidence. However, it is the lowest form of evidence. So I don't want to be dismissive and pretend as though individual clinicians' observations have no place in terms of generating knowledge. They do, but they are also most fraught for bias compared with the other forms of studies. All right. I also don't want to pretend that every question warrants a randomized controlled trial. There are naturally certain questions that don't. So this is just one example. This was published in the British Medical Journal a number of years ago. As you can tell pretty quickly through reading through it, it's satirical. But the reality, they said, okay, we want to do a systematic review. We want to do one of these grade recommendation type things that Rick described on this question of whether parachutes work. So we're going to go to the literature and we're going to find every randomized controlled trial published on this topic. And then they found that there were none. Okay? Why is that? I don't think I have to explain to you why that is. First of all, it's obvious what the effectiveness is based on just case series. Several people jumped out of an airplane. They all had parachutes on. They all survived. That's pretty good evidence in this case because it's not so complex, the question that's being addressed. But in medicine, rarely is the question this straightforward. Usually it's much more complex. Now, there are some that are straightforward. Many surgical questions are answered through case series. The patient had appendicitis. We wanted to know if we took the appendix out, if they would get better. We did that several times. Lo and behold, they got better every time. This case series approach does have its place in medicine, but most often it's much more complex to know the truth. And therefore, this approach of doing case series or just direct observation one after the other is ineffective. This is just one example, and I'm not trying to pick on any one person or any one topic, but I show you this because if you paid any attention to the news in the last 2 years, you probably saw this, okay? A physician, her name is not important for my presentation, said, had a widely publicized claim, this virus has a cure, it's called hydroxychloroquine. I've treated over 350 patients and had not one death. What is that? Well, that's a case series. Now, if she had published it and it had been peer-reviewed and been subjected to the rigors of the peer review process, then it would be considered a case series. It wasn't. It was just one person's claim. So it's actually even lower quality evidence than a case series. But nevertheless, if we take her at her word, and that's what I'm doing, this is what she observed. And therefore, it is a level of evidence. But there are numerous problems with this level of evidence. The biggest is that there was no control group. It could very well have been that had she seen another similar 350 patients and not given them hydroxychloroquine, not a single one of them had died. There's no way to determine, based on a series of patients like this, whether or not the treatment being scrutinized or questioned or evaluated actually resulted in the outcomes that are being attributed to that treatment. So, control groups are important. This was recognized as early as the time of Daniel. As far as I can tell, this is the first documentation of a clinical trial. Now, I'm pretty sure it wasn't a randomized clinical trial, so there's still some potential bias in this trial. But nevertheless, when Nebuchadnezzar said, "You're going to eat meat and wine," what did Daniel say? Now, Nebuchadnezzar, I'm making some assumptions here, but I think he thought, because it's the best diet for you, and that's what all my people do. And Daniel said, no, we have an alternative, and we think our alternative is better. But he didn't just say, and because we've done it, we know it's better. He said, let's compare. You do your thing, we'll do our thing, and then we'll come back together and we'll see who looks healthier. Now, there's some problems with this approach. It's not exactly the most rigorous, but it's better than saying, I saw 350 patients and they all improved, therefore my treatment works. Because at least there's a control group. At least they're comparing one treatment with another. And that's an important feature of randomized controlled trials. The first sort of modern, and when I say modern, we're talking about the mid-1700s here, application of a clinical trial with a control group, again, was not randomized, was James Lynn, who evaluated citrus for the treatment of scurvy. He said, there were a number of, actually, it turned out that his rationale for thinking that the treatment would work was completely wrong. But nevertheless, he said, I think that citrus may work for scurvy, but to know, I need to compare those who receive it to those who do not. So he implemented a clinical trial, and that led to the identification of the treatment of scurvy. Studies have bias. Research has bias. When I say research or bias, what I mean is systematic error. Now, this definition, which comes from Merriam-Webster, not exactly a clinical trials textbook, says that bias is systematic error introduced in the sampling or testing, so basically research, by selecting or encouraging one outcome or answer over the others. you can understand why that would be problematic. If you're trying to ascertain truth and then you introduce systematic error, whether knowingly or unknowingly, that could lead to the wrong conclusion. And even though Merriam-Webster says in selecting or encouraging, that doesn't actually mean it has to be intentional. Most often, I believe that bias is unintentional. Whoever's doing, whether it's a clinician making observations consecutively and they don't recognize the systematic error that occurs because of selection bias, or whether it's a scientist that designs a study that doesn't account for sources of bias, in most cases, bias is not intentional. So my goal is not to tell you that everyone who's not doing randomized controlled trials is just, intentionally leading to misinformation. In fact, I published more randomized controlled trials than I have not. I actually meant to tell you during my disclosure slide when I said that I have academic interest. I've published, I think, around 16 randomized controlled trials. Probably doesn't sound like very much, but these things take forever to do. And actually, earlier when we were discussing the speed with which the responses came and the information was developed, I didn't want to slow us down because I wanted to get to lunch. But I was thinking, are you kidding me? This has been an amazing success. It is phenomenal how fast the knowledge was obtained in the last 2 years. It's without precedent. Never before have we seen, and correct me if I'm wrong, you're the vaccinologist, never before have we seen vaccines develop so quickly. Never before have we seen clinical trials identify novel treatments so quickly. In the history of medicine, we have never been as fast as we were in the last 2 years. That said, could we do better? Yes, absolutely we could. But I think we should recognize and acknowledge sort of the difficulty with which this work is conducted and how complex it is. So I got a little bit off track. I was going to tell you. So I think I have around 160 publications, and about 16 of those are randomized controlled trials. Most of the rest are observational studies. So what I'm going to do in the next few slides is poke holes in the approach to non-randomized controlled trials. I'm talking to myself here. So I'm a scientist that does non-randomized controlled trials as well as randomized controlled trials. And there's an important place for that type of research, but we have to acknowledge the limitations and the reasons that we do randomized controlled trials. So one of the most important limitations is selection bias. Selection bias is essentially what occurs, it's measurement error or systematic error that occurs because a group of participants that are analyzed or observed, and we call that the sample, is not representative of the population intended to be studied. So some important terminology here. The sample is the group of people that are in the study. The population is the larger group that they're supposed to represent. So if we want to know the effectiveness of vaccines for adults, then the population is all adults in the world. It's a massive population. The sample are the people that are actually in the clinical trials. Selection bias, or not just clinical trials, studies, whatever that study may be, the case series, the observational study, selection bias occurs when the people in the study are not representative of the larger population. So the 250 or 350, I've now forgot, patients who were treated with hydroxychloroquine and not a single one died, were those people representative of all patients with COVID? Absolutely not. And if that study had been subjected to peer review, that's one of the things that would have been highlighted, is that there's a tremendous amount of selection bias. If you're an outpatient clinician, do you know who you're going to see for COVID? The people that are doing well enough to go to their outpatient clinician. If you're a critical care physician, do you know who you're going to see for COVID? The people who are dying. And most of them wait until they're on death's door before they decide to go get medical care. So there's a huge amount of selection bias that leads to the observation that, well, all of the patients who walked into my clinic and asked for a prescription did fine. Of course they did. They wouldn't have shown up at your clinic's door if they weren't doing fine. Okay? So selection bias is a huge problem, and that's one of the problems addressed by randomized controlled trials. I'm not going to go through the details of the types because I'm realizing already that I'm taking more time than I intended to. Information is another big problem. This occurs from measurement error. You can have measurement error in terms of ascertaining the outcome, of identifying the exposure, and in terms of the timing of the events. There are all sorts of sources of measurement error, which lead to information bias. And these sources of error are addressed for the most part, but not perfectly, in randomized controlled trials. All right, so now we're stepping up. And we went from case studies, and I gave you one example. Now we're up to the next level of evidence, still way below in terms of rigor, a randomized controlled trial. This was a retrospective analysis. So what they said was, okay, we're going to analyze all the people that have been admitted to our hospital from March 10th to May the 2nd, 2020 with COVID. And then we're going to compare those who got hydroxychloroquine with those who did not. And the results of this study were that they found that patients who got hydroxychloroquine did better than those who did not. This is better than the case series, but not much better. There are methods by which to try to counter the effects of selection bias, which is still a huge issue here. I think my explanation of selection bias for the clinic was pretty straightforward. It's a little bit more complex, but selection bias occurs the same way in routine clinical care. In the hospital, there are certain reasons that we give patients certain drugs. Concern for side effect, anticipated benefit. If the clinicians that were treating patients at Henry Ford Hospital with hydroxychloroquine from March to May of 2020, knowingly or unknowingly gave the drug to patients who looked a little bit better, which is very, very likely that they did, because you would be more afraid of side effects in the patients who are already doing poorly, and you would be a little bit more nihilistic, unfortunately, in the patients who are already doing poorly, and therefore you'd be less likely to give them this drug, and you would target the patients who looked better in the first place with the drug. Then you're going to introduce selection bias. So I think I've shown a few examples here. I'm not trying to pick on hydroxychloroquine, but it's an easy one to highlight because there are easy examples to find of case series of retrospective and prospective cohort studies and of randomized controlled trials. With all that in mind, how do randomized controlled trials work? So hopefully I've explained some of the problems of non-randomized studies. How does randomization and the design of a randomized controlled trial address that? So there are two processes, and this is the slide that my daughter said, Dad, that should have been early on. There are two processes in a randomized controlled trial that distinguish it from other types of research. You think it was just one process, number one, but it's actually the second is just as important as the first. So the first process is a randomization. It sounds a little complex. Assignment of participants to the intervention is random, and actually, when we implement this, it can be quite complex because there are all sorts of details in terms of how to do that in the most rigorous way. But conceptually, the idea is just if you have a patient who's going to be in a trial and there are two possible treatments you're comparing, let's say hydroxychloroquine versus placebo, you flip a coin. For that individual patient, you just flip a coin, and that's how you find out, are they going to get in the hydroxychloroquine group or the placebo group? There's another really important part, though, and that is that the allocation is concealed. What do I mean by that? What I mean is that the intervention assignment, so in that example, the determination of whether or not they get hydroxychloroquine or placebo is not revealed prior to making that assignment, okay? So in the flip of the coin, if you say beforehand, okay, we're going to flip it, heads hydroxychloroquine, tails placebo, you don't know at the time that you decide heads is hydroxychloroquine and tails is placebo, which side is going to end up face up, right, when you flip it. Let me give you an alternative example. If you say, okay, we're going to do a trial, and every patient that gets admitted to our ICU on an odd day of the month is going to get hydroxychloroquine, and every day that gets admitted on an even day is going to get placebo, you might think that's random. And actually, in some senses it is, although there are some ways that you could fudge things to get around the randomization. But what is absolutely not the case is that the allocation is concealed. You know before you recruit Mr. Brown or Mrs. Smith into your trial that because it's the 20th of the month, they're going to get a certain treatment. I've already forgotten whether hydroxychloroquine or placebo is the even days. But the point is, you know before you bring them in what group they're going to be in. That is not a randomized controlled trial, and that opens you up for potential bias. So to be a true randomized controlled trial, you have to have both the randomization procedure and allocation concealment. If you're interested about how these are done, ask me afterwards. I'm happy to explain the details. So the first true randomized controlled trial was conducted in the United Kingdom in the 1940s, and they were evaluating the effectiveness or the efficacy and safety of streptomycin, a new antibiotic at the time, for the treatment of pulmonary tuberculosis. These are children with pulmonary tuberculosis in the 40s. There were so many of them. Now, this was not a new problem in the 40s, right? This had gone back decades and centuries more. But now they have this antibiotic. And there was good, promising animal model data to suggest that the streptomycin would effectively treat pulmonary tuberculosis. You know what animals were used in that study? Guinea pigs. So I don't know for a fact, but I think that this the use of guinea pigs in this preclinical model was maybe the origin of the term, I don't want to be a guinea pig. So they had guinea pigs who improved dramatically after being treated with streptomycin, and those who they had a control group in that animal model study, the guinea pigs that did not get streptomycin died of pulmonary tuberculosis. So the United Kingdom did a clinical trial. It wasn't a large one, but it was randomized. And they actually, in this case, they randomized for two reasons. And this is the first time this was done. One reason was because it was more rigorous. And I think I've already explained to you why that would be the case. So they had a brilliant statistician working with the group that designed this clinical trial that said, you know, this would be more rigorous if we randomized rather than use some other method of assigning patients to their treatment. The other reason they randomized is because they decided it was the most ethical thing to do, because they didn't have enough streptomycin to treat everybody. Now, even if they did, they wouldn't have determined that they should, because they didn't yet know that the drug was effective. But as we all know from the last 2 years and many other years, society doesn't wait to find out if rigorous data supports a treatment. They just decide on their own what's effective, and then they ask for it. And so they were facing the same problem. There were people that knew, oh, streptomycin, it might be effective. It helped guinea pigs. Give it to me. And they didn't have enough. Even if they wanted to, they didn't have enough. And so they said, okay, how are we going to decide which patients that agree to be in the clinical trial get the drug, which is what they want, and which ones don't get the drug? And they decided that the most ethical way was randomization. So this is the first randomized control trial. What are the benefits? Well, I think I've explained this, but I'll just reemphasize. First and foremost, it eliminates selection bias. So you're not choosing which patients to treat based on how they look, based on their propensity for side effects, based on your expected outcome, based on who your brother is. You're not choosing, you're simply randomly assigning, and this eliminates selection bias. It also facilitates blinding, or sometimes called masking. I haven't talked about this, but briefly, the idea here is that there are a number of reasons we might want to actually not know as we're doing the clinical trial which treatment a research participant is receiving. For example, it would help us to avoid information bias that I mentioned earlier. If we knew when we were measuring the outcomes or the side effects in question, then we might either knowingly or unknowingly collect information in a way that favors one group or another. By blinding or masking, you don't know when you're collecting the information, and not even the participant knows when they're reporting to you their symptoms and how they're doing, whether or not they're actually getting treatment A or treatment B. No one is aware, and you don't find out until the end of the trial. Now, briefly, you might think, well, gee, that sounds kind of dangerous. Isn't it a good idea to know what treatment someone's receiving, especially if it's some new experimental treatment? There are safeguards in place to protect against that, and it's possible to do what we call break the blind. In other words, to find out in the middle of the trial which treatment a patient received if we need to in the interest of safety. But in most cases, you don't know. And the only type of clinical trial that allows for this is a randomized controlled trial. Similarly, that would also prevent confirmation bias. You know, if the person, I think I've explained this, if the person who's doing the measurement already thinks they know what the right answer is, they may be biased in terms of how they collect the information. And then finally, and I'm not going to spend any time on this at all, other than just to say randomization permits the use of probability theory when analyzing results. So this facilitates some of the statistical nature of the analysis of the clinical trial. All right, so I've given you examples of case series. I've given you examples of observational studies. This was the first high-quality, large, randomized controlled trial that was published examining hydroxychloroquine as a treatment for hospitalized patients with COVID compared with placebo. This is actually quite large for a trial of its kind. It's not nearly as large as some of the vaccination trials, but for this type of trial, it was very large. It was done in the United Kingdom. It had no relationship whatsoever to industry, and it was actually done very quickly, which was very impressive. And what they found is that patients who were in the hydroxychloroquine group statistically had the same outcomes or similar outcomes to those who were in the control group. And I say statistically because, you know, if you look here at the confidence interval, the rate ratio for death included 1.0, which would mean they had the exact same rate ratio, or they have the exact same rate of mortality. If you want to know, though, if you wanted to sort of place your bets, like what's the most likely effect, what does the trial think is the effect, you would say it's this: 9% more likely to die if you receive hydroxychloroquine than if you did not. Now, the statistical approaches we use, the truest interpretation of this result is that they were the same. This suggests that there may have been harm, but what it absolutely does not suggest is that there's any benefits whatsoever. This was not the only trial. There were lots of other trials, and so then when someone does a grade recommendation style analysis, they conclude that hydroxychloroquine has no beneficial effect for patients with acute COVID. This is true not just for hospitalized patients, which were in this trial, but for patients who were treated in the outpatient setting. It was also studied as a potential sort of prophylactic treatment. You get exposed, then you take hydroxychloroquine. Does that then prevent you turning positive? Across the board, it didn't matter how hydroxychloroquine was examined. If it was a randomized controlled trial, there were no beneficial effects that were identified. And in the interest of disclosure, I was part of at least one clinical trial, and right there, you can see it takes a lot of people to do these. This was an international clinical trial conducted in the United States and a number of countries in Europe and in Australia and New Zealand. We looked at actually multiple treatments in this trial. The title sort of implies that we combined these antiretrovirals, lopinavir, ritonavir, with hydroxychloroquine. That was not what happened. We looked at either or and also compared with no treatment. And across the board, there was no benefit for hydroxychloroquine. Interestingly enough, we found that lopinavir, ritonavir was harmful in this trial. So I want to sum up in terms of comparing randomized controlled trials with non-randomized clinical studies by looking at this from the perspective of the research participant. Okay, I think I've primarily described it to you from the perspective of being the scientist and trying to discern the truth. But I think it's also important for us to think about this from the perspective of the participant. So I've just given you a number of potential features of the participant's experience, depending on whether or not they were in a case series, and this could either be sort of a non-published case series at their own clinician's office, or a published case series, a non-randomized trial. So that would be a trial that did have a control group, but they didn't randomize. And then, of course, the randomized control trial that I've been describing. So access to novel treatments doesn't happen outside of controlled studies for a number of reasons. So you get a negative in the case series aspect. That can definitely happen in a non-randomized trial, but it's most likely to happen in a randomized control trial. Now, whether or not that's a good thing or a bad thing depends on whether or not that treatment was good or bad, and you don't know until you do the test. But this is a very common reason that patients with advanced cancer end up in randomized controlled trials, because they want access. They've already received the standard treatments, they've not gotten better, and they want access to the novel treatments. The way that they get that access is by participating in a randomized controlled trial. But it's their choice, and the potential adverse effects are described as well as the potential benefits. So I don't want you to think anyone's led to believe, like, look, you're going to get better if you just get in this trial, because there are lengthy, rigorous steps taken to ensure that no one thinks I'm guaranteed to get better as part of this trial. Increased chance of receiving the most effective treatment. That really only happens in the context of an adaptive randomized controlled trial. That's a feature of randomized controlled trials that I haven't described, and actually was very uncommon in many areas, mostly was used in cancer treatments, but it actually allowed, remember when I said you flip that coin and it's always going to be 50-50 chance? Well, an adaptive randomized controlled trial, you can actually have a design that allows for the data that's accrued during the trial to inform the probability that the patient receives treatment A or B, or if there are more treatments, C, D, E, and so forth. So you can actually build into the trial in a scientifically rigorous way a method that allows you not only to say, here are the risks, here are the benefits, and you might get an effective treatment, but to say you're more likely to get the effective treatment than you are the non-effective treatment, because that changes over time as the data update. Independent oversight. I'm going to actually explain this in more detail in the last portion of my talk if I don't run out of time. But that really occurs most regularly in a randomized controlled trial. Enhanced systematic safety monitoring, again, occurs most regularly in a randomized controlled trial. It does not occur at all outside of a clinical research setting. There's no systematic safety monitoring. It may be that your doctor who gives you a treatment says, well, maybe we should check this lab to look for a side effect, but it's by no means systematic and it's not enhanced. And then lastly, contribution to the care of future patients. So this sometimes occurs with case series in select cases, but really the highest likelihood of contributing to the care of future patients because you were part of a trial that led to new knowledge is to be in a randomized controlled trial. All right, so the last part of my talk, and I actually don't remember, David, what time I started.
About 5 minutes.
5 minutes, goodness gracious. Okay, this is the hardest part. Are randomized controlled trials trustworthy? Okay, I'm going to try to go quickly. So the limitations of trials are by no means exhaustively listed here, but I tried to list some of the most important. The first is human error, which might be unintentional, or fraud, which is intentional. This can certainly occur in the context of a randomized controlled trial. The second is expense. They are not cheap. The third, sampling bias, and that occurs when the sample, that's the people in the trial, end up not reflecting the characteristics of the population you're trying to study. That can happen. And then heterogeneity of treatment effect. Ask me afterwards if that sounds interesting. All right, so why should I be concerned about human error or fraud? Well, because human beings make errors and they commit fraudulent acts. This is a fact that we all know is true. And it occurs regardless of whether you're doing a clinical trial or a case series or any other endeavor. So, and here's just one example. This was a well-publicized instance of fraud. It was not in the setting of a randomized controlled trial. It would have been caught if it were, for the reasons I'm going to show you in the next few slides. But the data that were used for a number of non-controlled or non-randomized, rather, observational studies early in COVID turned out were fabricated. They were just made up. And eventually, it was identified mostly through the process of peer review. But this can occur in a randomized controlled trial as well. This example is from non-randomized trials. So what are the safeguards? Well, there are a number of safeguards, and I don't think I've shown you all of them, but I want to highlight the most important. Okay, regulatory review, I'll describe in more detail. Registration, I'll show you what that looks like. Monitoring, both internal and external. Data sharing, peer review, and disclosures. Okay. So first of all, regulatory review of randomized controlled trials. to do a randomized controlled trial in the United States for involving a drug that is not currently known to be effective for the condition you're treating, especially a new drug, for example, or a new vaccine, you have to, first of all, get FDA approval. Now, whether or not you believe or trust the FDA, I'm not here to argue, but my point is there are people who are supposed to be experts, and most of them at the FDA are, who are looking at your protocol to determine whether or not it's well designed and is safe. Second, you have to have a data and safety monitoring board. This is a group of independent experts that are not part of the FDA, not part of the government, but are peers who have no existing relationship with you that evaluate your protocol and then over the course of the trial actually evaluate the data to identify safety problems or other issues that could compromise the integrity of the trial. And then lastly, locally, there are institutional review boards. Again, a group of peers that are designed to ensure that the OHRP regulations for human subjects research in the United States are followed in the context of this trial. Registration is another important safeguard. So gone are the days when you could say it in a trial, say you specified that you're going to test the effect of treatment A on B outcome, and then cherry pick the results depending on what the data you have. Can't do that anymore, and the reason is because there are federal requirements punishable by prison time that require that you specify on this publicly available website what you're going to do, how you're going to do it, and therefore you cannot change or cherry pick the results afterwards without people knowing that you did that. Journals will not publish trials that have not been registered. So this is just one example of a randomized controlled trial for hydroxychloroquine. It's not the one I showed you earlier because that was done in the UK. They have their own system for registration. But this one, you can see they first posted on April 3rd, 2020, pretty early. And then the results were posted in March 17th, 2021, a little under a year later, which is really fast, really, really fast. And then you can also actually even see every single time they made changes and what those changes were. So this is all tracked. And as I said, not only do the journals hold you accountable to this, but the federal government does. So you cannot cherry pick. You can't change the story after you get the data. There's monitoring. I've already described what the DSMB looks like. There, in addition, there are audits, external audits, where someone either from the sponsor, like the NIH, or from your local IRB will come in and look at your data, look at your documentation, ensure that you're following all the guidelines, show me your consent forms, and so forth. And then you make the data available. If it's federally funded, you have to make the data available. This is a clinical trial that I and others did a number of years ago. It's not related to COVID. It was published in 2018. But you can see here that on this publicly accessible website, anybody who wants to can go to the data, download it, and analyze it. Peer review is another important part of the process. I think most of you are familiar with that in your fields. We have peer review in our fields. This is actually a very fast timeline for peer review. This was the trial I showed you earlier that I was involved in with hydroxychloroquine. We submitted the paper on April 11th, 2021, and they accepted it after several revisions in that, 6 weeks timeline, and then it was published online 2 months, a month and a half later. And then lastly, disclosures. So the same type of disclosure that I showed you at the beginning of this presentation, that's made by everybody that's involved in a clinical trial. They have to disclose not only to their sponsors, to their local institutions, who all have a conflict of interest evaluation process, but then also to the general public. So every single trial that I publish, you can go on the website of the journal that published it, and you can download my disclosures that were true at that time. I'm going to skip the next couple slides because I think that I'm out of time, and maybe we have time for a couple questions.
If you're done, we can ask questions. Are you done?
Okay, I'm done. I'm done. That's it.
Okay, there's one right here. You need to repeat the question.
Okay. if I understand them, I'll repeat it.
So I'm a little confused sometimes when I'm reading through medical work. It seems like my impression is that a lot of it is just the output of SAS or SPSS. And they just like spitting out what this computer program has spit out. So they get all these things, and I'm not even confident that they know what the black box is spitting out, and that they understand what these things are doing. They understand the controls, what's being controlled for, but oftentimes it's like 8 or 9 things being controlled for, and I'm not convinced that linear correlation that they ended up with well outside, tiny p-values isn't, for example, dragged by one data point.
Okay.
Do you have confidence in that? I mean, what level of confidence do I have reading a medical publication when they only give that output?
Right. Okay. So the question, I'll try to summarize. What I heard you say is that statistical analyses that are used to analyze biomedical research are complex, and therefore, how does the reader or the consumer of this output have confidence that these analyses were done correctly? Is that right? Okay. That is a good question, and there are a number of ways I would answer it. Number one is that the data are made available, and so the analyses can be repeated if there's anything suspicious. And that has happened, and that actually has led to detection of fraud. It's pretty rare because I think most people know that they're not going to get away with it. But it is an option to actually get the data, redo the analyses, and confirm. Number two, the statistical analyses in randomized trials are actually typically one of the more straightforward analyses that are done in biomedical research. So like the examples, well, what I heard you allude to, if I understood correctly, were the types of analyses that are typically done in observational studies, where because you didn't use randomization, there's a higher level of concern for confounding from sources of bias, and therefore you have to use complex statistical techniques like regression modeling to try to account for that. Now, we're getting probably way too into the nitty-gritty here for me to say this. There are actually really good reasons that randomized controlled trial data should also be used or analyzed with regression analysis. There's some statistical reasons that that's the case. But at its simplest level, a randomized controlled trial is designed so that you can just take one group, take the other group, and just tally up the difference in your dichotomous outcome. How many died in this group? How many died in that group? You could do a simple chi-square test. Anybody could do that online. If you look at an abstract for the paper and they say, this number died out of this many treated, this number died out of this many treated, any one of us could actually just go and replicate that analysis in 5 seconds. So randomized controlled trials actually lead to less complex analysis than other forms of biomedical research. Another part of the answer is that the software that's used is highly reliable. Most of it, like the examples you gave, are actually industry-created software, and not everyone uses industry-created software. A lot of people use R, which is open source software, but nevertheless, the statistical packages are very reliable. It's not a black box. You know exactly what's happening. Now, whether or not you would want to manually do that yourself, no one would do that. But if they wanted to, they actually could. Like the calculations for a student's t-test or a chi-square or even linear regression, logistic regression, they could be done manually. They're not black box analyses. There are studies that use more black box analyses, machine learning, as we talked about a little bit at lunch, but not for this type of research.
I think my sense is that it's a black box to a lot of the research, not that it's a black box.
Yeah. Okay, so then that point is, well, how do you deal with the fact that some researchers just don't know what they're doing?
Well, that's what
right, and that's true.
The question is that if they just dump what the computer program gave out, how do I know they understood that? Yeah,
and so the answer to that is several of the things that I mentioned, but I didn't explicitly specify that it would help in this instance. Okay, peer review is a big part of that. You can pretty quickly detect that people didn't know what they were doing in the peer review process. And number two is the registration and the other regulatory assessments. So you have to have a pre-specified protocol for a randomized controlled trial. You can't do a trial and then decide later to report it. You have to specify what you're going to do up front, and that's reviewed. And if you don't know what you're doing when you outline the protocol, you're going to be detected and you're going to be corrected.
So here,
Just to add on to that, and as part of peer review that Tim talked about, all medical papers for an RCT would have a section in the paper called discussion, where you would be able to read the investigator's understanding of the results of that clinical trial and be able to judge, do they get this or do they not have it right?
Right. And I think that's actually much less common in randomized trials because of all of the review before they do it. But it is very common in non-randomized observational studies. You know, someone collects a bunch of data, they do their analysis, they don't understand it, and then they try to publish it, and the peer review process says, oh, not so fast. Here are the problems.
over here.
Super question. One is, I'm guessing you heard the replicability crisis in science at large.
Yeah.
Is it fair to say that RCTs have been fairly immune to that, or?
Well, I wouldn't use the word immune. That's a really strong word when you're in the context with which you use that word. Yeah. So the question was, are randomized controlled trials immune to the reproducibility crisis? Okay. So as I understand it,
I need to find reproducibility crisis first.
that's what I was going to do. So as I understand it, this was really first. It first received a lot of publicity in sort of the social sciences, psychology, sociology, where I fully admit I'm not in that field, so I may be misrepresenting based on what the media has said, which is a common occurrence, right? The media gets it wrong sometimes. But as I understand it, there are a lot of conclusions that were made based on psychology studies. And it's like, okay, it was shown once, it must be the truth. And then eventually people started deciding, well, let's and I think in that field, actually, there are a tremendous amount of pressure to prevent people from repeating studies. And when someone finally got over that pressure and tried to reproduce some of the studies, they found that the results were not reproducible. So in other words, well, we thought everything was well controlled and we had a solid, rigorous conclusion, but it turned out that we couldn't reproduce it. Therefore, maybe it was never right in the first place. The answer to your question is a little bit, but not perfectly. Like randomized controlled trials do get repeated, and sometimes they are reproducible, and other times they're not. And actually, this is the reason that the process that Rick described of using a rigorous evaluation of multiple trials together is so important, because the results might not be identical. And, you know, he showed us one figure that was a forest plot where it showed like here's a whole bunch of trials, 10 trials or something, all examining the efficacy of a particular vaccine, and the results were not identical. So in that sense, they weren't reproduced, but they were actually all very similar. And the lack of precision was primarily because of what I mentioned earlier, the sampling bias. None of the groups or samples in individual trials is a perfect representation of the larger population that you're trying to understand. But the more trials you do, you can take that data together and get a more accurate assessment of the truth.
Can I just add one thing? So for drug approval in the U.S., right, the FDA, following the U.S. Code of Federal Regulations, this is why it requires 2 randomized controlled trials. Now, there are exceptions, including emergency use authorization, but that's one reason why it's codified in our law, that it takes 2 randomized controlled trials to establish efficacy.
Right.
Okay, are there any from the internet? Okay, any other comments or questions? I can add. Okay, did you also?
I actually.
Okay, well, we'll take 2 more.
So,
Just a question about peer review. I think sometimes for people outside the medical or scientific community, it feels like the whole thing is this big industry.
Yeah.
But as I think about it more, peer review means that people are reviewing the work that themselves aren't going to benefit from the work being a success. In fact, given the competition for research dollars, they might actually be really highly motivated to find a problem with someone else's work. Rather than being, so I just wonder if you could comment on that a little bit, that the reality of being inside that medical community as people evaluate each other's work, how does that affect the process?
Can you summarize that as well?
Yeah, so the question was regarding the approach to peer review, the fact that those people are peers, meaning they're experts in the field, but they're not part of the study team, so they're external. Does that make them more likely to give an unbiased assessment, or, as the question pointed out, potentially even a sort of a more ruthless assessment because they might view the authors of the study that they're reviewing as competitors? I definitely think the intention is that it's an unbiased assessment. And the authors do not get to pick their peer reviewers. The peer reviewers are picked by people like Greg, the journal editors. So when the journal editors receive a submission, they look at the manuscript. First, they decide, is this even appropriate for my journal? Second, is it worthy of spending more time on? And then third, if it is, who am I going to ask based on who I know is, number one, an expert in this field, and number two, not connected to this group of authors, to then give an unbiased peer review. So the intention is definitely that they're unbiased. My general experience is that, if anything, you're probably or you're right, that they can sometimes be overly scrutinized. I don't know whether or not that's because people view each other as competitors. I think mostly it's just because as scientists, we're naturally sort of inquisitive, we're naturally, intrigued by the possibilities, and we want to know, show me the proof. We want to think about every possible explanation, and so we're rigorous. And so it can be a demanding process. I have had, of my 160 some odd papers, I would say a third of them, I spent more words responding to the peer reviewers than I did actually writing the manuscript. So, most manuscripts are like 3,000 words. You have to write a point-by-point response to the peer reviewers. And sometimes, depending on the complexity of the study you're presenting, you actually write, 20 pages of response. So it's a very rigorous process.
Is that it then? Chairman.
Just going to raise the question. Ask the question, are RCTs trustworthy? I think one interpretation of that is, are they a good guide for what we should do? Those are different questions.
They are different questions. You're right.
You can speak to that, how somebody might use an RCT finding a very narrow piece of information. It had this effect on this particular outcome, and conflate it with C, policy needs to be X.
Yes. Okay. So for those online, the question is, I'm claiming that RCTs in general, not specific RCTs, but across the board, are trustworthy. Is that the same as saying that they actually should be the source of policy making? And I would say it depends. It depends on how the trial was designed, and how the outcomes that were assessed relate to the policy that's trying to be made. It's not just a knee-jerk reaction, absolutely. And actually, I think I'll end with this. I'm going to take you back to that idea of doing a randomized controlled trial to test whether parachutes are efficacious. All right, so a follow-up study, again, satirical, published in the British Medical Journal, said this: we did a randomized controlled trial to test whether parachutes were efficacious, and we found that they were not. Out of 10 patients or 10 participants who were given a parachute, 0 died. Out of 10 who were not given a parachute, 0 died. And therefore, our trial concludes parachutes do not prevent death for patients who are jumping out of the airplane. And in the discussion, it said, we're concerned that the results of our randomized control trial may not be generalizable to all settings in which a parachute would be used. In our particular case, we were unable to recruit anyone who was willing to jump out of an airplane that was not sitting on the ground, and therefore our results are only applicable to the use of a parachute when jumping 3 feet out of an airplane that's sitting on the ground. So you're absolutely right. There are many, many contexts in which someone may point to a specific randomized controlled trial and say, see, this proves my point, we should do things this way. And they could be completely off base because the details of the trial are important. It matters what the sample was. It matters what the characteristics of the people were that were involved. It matters what the intervention was and how it was delivered and where it was delivered. And it matters what the outcomes were that were assessed. All of those things are important. And again, I keep pointing back to Rick and Greg's presentations because you guys did an amazing job this morning. Rick pointed this out when he talked about the PICO approach. You know, the PICO approach to evaluating randomized controlled trials as a group and then determining recommendations, clinical recommendations based on those, requires that you ask, what was the population that was included? Was it the population that we're trying to make a recommendation about, or was it something entirely different? What was the exact intervention? Because if the intervention was a little bit like what we're looking at, but not this exact same, we're not going to include it in our evaluation. What was the comparator? Because it matters. Or you can comparing to placebo? Were you comparing to some current standard of care? Because it matters. And then lastly, what were the outcomes? Because if the outcomes that we care most about wasn't measured, then we're not going to pretend that we know how this intervention is going to affect that outcome when it wasn't actually measured. So all of those details are really important. And you're absolutely right. That has to be considered.
Can I just talk about the PICO again? And maybe I'm a PICO enthusiast, but ivermectin has gotten a lot of press. And a lot of Christians have said ivermectin works. And indeed, there are randomized trials that show ivermectin works. But when put to a recent study that I saw that put some of the studies together, the ivermectin worked in the countries or the areas of the world where strongyloides, a parasite, was prevalent. And so the conclusion of the meta-analysis is that if you live in a country where parasites like strongyloides are present, and you likely then have COVID and strongyloides infection both, you will be helped by ivermectin because it will take care of the strongyloides. But if you are in a country where there is no parasite, the ivermectin will do you no good. And therefore, why is there a difference in the ivermectin results? Well, it's because of the presence of parasites like strongyloides or whatever, which gets back to PICO. What's your population and what is the intervention that you're trying to do? Sorry.
Okay, well, let's thank Tim again.